My name is Brigitte Gomperts. I'm at UCLA. And thank you so much for this opportunity to tell you about some of the work that we've been doing, looking at the airway epithelium that's exposed to both cigarette smoke and SARS-CoV-2, which is the virus that causes COVID-19. My lab studies lung repair and regeneration after injury, and we've done some studies after tobacco smoke injury before, but this was our first foray into looking at viral infections. But the pandemic already spurred some wonderful collaborations here at UCLA that I'll tell you about. So just by way of background, I just want to tell you a little bit about the lungs because they're very complex and very important organs. And when we think about the lungs, I actually start thinking about the nose and mouth as the upper airways, which then extend through the throat down into the trachea, which is the windpipe. And this part of the lungs is directly in contact with the environment. And so it plays this very vital role in host defense. And the way that it does this is it generates a whole lot of mucus that traps the bacteria, the viruses, pollution, cigarette smoke, everything that we're breathing in. And then there are these cells that are highly specialized that have hair-like projections that beat in one direction. And they move the mucus with all these trapped particles up and out of the airways. And then if we move further down, we go through the bronchial structures, which conduct gas, and eventually we get to these alveolar structures, which are these grape-like structures shown here. And this is really the unit where the gas exchange occurs, where oxygen enters the body and carbon dioxide is breathed out. And there's this very large surface area so that we just are able to do this process through diffusion. And while tobacco smoke and of course SARS-CoV-2 can infect anywhere along the respiratory tract, and tobacco smoke can affect all of these different regions, today I'm really going to focus on these upper airways, particularly in this tracheal airway, to describe some of the effects that we've been seeing of this interaction between tobacco smoke and SARS-CoV-2. So if we talk a little bit more about mucocillary clearance, as I mentioned before, this is this vital host defense mechanism. And there are these highly specialized cells within the pseudostratified epithelium, which is this barrier lining of our tracheas. And there are these specific cell types that I'll briefly mention. So there are these basal cells, which are shown here in orange, which are really the stem cells for the airway. And that just means that they make more of themselves. And then they can also differentiate into the specialized cells of the airway, which are very important at making the mucus, which is here, this goblet cell, and then the ciliated cells, which have got these hair-like projections that beat unilaterally. And this is just a movie showing how these hairs beat and how they can move the mucus and the particles up and out of the airways. And so here I'm showing you an actual cross-section through a human airway with all of these different cell types that I mentioned before and how they're layered within this epithelium. And one of the major advances for us in the lab to be able to model this airway epithelium came about a decade ago when some investigators figured out that you could actually take these primary human cells. So what we actually do is we get in these human samples of tracheas, we're actually able to digest and strip away all of the surface epithelium, and then we can isolate out just these basal cells, these stem cells shown here in purple. And if we layer them on a membrane and culture them in media, and the media will be on the top and on the bottom with all the nutrients that the cells need, they will grow very nicely and form a monolayer. And then if we take the media from the top and we just have air at the top and the media with all the nutrients in the bottom, these cells actually have these programs that will allow them to differentiate into these mucus-producing cells and these ciliated cells. And so they really beautifully reproduce what we see in the body. And this is what it looks like when we assess these cultures in the dish. And so in green here are the ciliated cells, lots and lots of these hair-like projections, which form almost like a carpet. And we're staining here in green for acetylated tubulin, the marker of the ciliated cells. We can look at mucus production, which is this pink color. All of the pink that you see is actual mucus in these cultures. And then in the white, I'm showing you keratin 5, which is a marker of the stem cells. And so these are pretty tricky things to do, but we were able to do it in the dish now. And it's thanks to my wonderful team, Arunima Prakayastha, Chandani Sen, and Abdo Dara, who figured out the conditions to grow these really well. So what's really nice about the fact that these cultures have air on the top of them is that we can then expose them to a number of different things that you would think about people would be exposed to if they're breathing into the airways. And so we have developed this system, which is really a closed box that we can put the little cultures into in their dish. I think you can see it through here. And then through a series of vacuum pumps, we can directly hook up something like a research grade cigarette. We can also do the same thing for an e-cigarette. And we can then basically through an Arduino based remote controlled system, we can actually program in the duration of the puff, how long the smoke will sit, the cigarette smoke will sit in the chamber, and when it will be taken out. And so we have this controlled system. And so what we've studied over time is we've realized that we can expose the cultures to cigarette smoke for just 3 minutes a day for 4 days in order to get an injury cells, but not enough that they're all die off, because if we smoke for too long, then the whole culture system dies off. And so this is the schematic of what we did for all of our studies, is that we seeded the stem cells. And then after 5 days, when they formed a layer, we took them to that air-liquid interface on the transwell system. And then we grew them and allowed them to differentiate to the mucus cells and the ciliated cells, just like the epithelium that you would see in the trachea. And then we did the 4 days of smoking. And then the next day, we infected them with the virus, the SARS-CoV-2 virus. And then we left that on. We left the viral infection going for 3 days, and then we deactivated the virus. And of course, all of this, I should say, was done in BSL-3, highly controlled conditions. But once the virus was deactivated, we were able to get these cultures back to do our downstream analyses. And so this is what the data look like. And so the first thing that struck us when we looked at the cultures was how different they looked. So here we're staining for the spike protein, which is a protein found on the SARS-CoV-2 virus. And this is cultures that had no cigarette smoke, but did get the virus. And you can see that we see these patches. It's actually remarkable that this epithelium, even though we grow it in the dish, has got a very good innate immune system. It fights off the viral infection. So we really only see about 1 to 5% of the cells being infected, but they tend to be in these little clusters. But for every single culture that we've done, and now we've done, probably 50, every single time we do it, we see that in the presence of cigarette smoke, we see more clusters, and the clusters tend to be larger. And we can actually use PCR to analyze the amount of viral load, like how much virus there is in the cultures. And we see consistently that in smokers, there is always a higher amount, particularly at day 2. But after day 2, the cultures start to fall apart and some of the cells die and move out. So after that, our PCR is not very accurate. Oh, and then I should just also mention that we quantified the percentage of infected cells. This is in no cigarette smoke, and here was cigarette smoke. And you can see that there's this very significant difference between the cultures. And this was all performed by Gus Garcia, who's a very talented researcher in Vaiti Arumugoswamy's lab. And Vaiti is the virologist we've been collaborating with to do these infections. And so we wanted to know more about what the virus was doing to our little cultures. And so here you can see we have these 4 groups of no cigarette smoke, no virus, cigarette smoke, but no virus, so just cigarette smoke alone. Here is just virus alone without the cigarette smoke. And here is both cigarette smoke and virus. And we were looking to see with these ciliated cells, which have got those hair-like projections, how those do in the setting of these different treatments. And we did see patchy loss of the ciliated cells just with cigarette smoke alone, but because it was only in some areas, it actually ended up not being statistically significant. In the setting of just virus alone, there was no change in the number of ciliated cells, and there was a trend again to some more loss of cilia when there was cigarette smoke and virus, but it wasn't significant. What we did see in the setting of looking for mucus is that cigarette smoke, I guess this is no surprise to anybody, greatly increased the mucus production. This was significant, but the virus, interesting enough, did not induce mucus production, but there was a trend towards it when we looked at it in the setting of cigarette smoke with virus. And then when we looked at the stem cells shown here in pink, we saw that cigarette smoke greatly induces these stem cells. Obviously, they perceive that there's an injury and they start to repair. But the virus alone did not induce any kind of repair response. It didn't seem like the airway epithelium was recognizing that there was any kind of injury, which we think is really interesting. And then, of course, there was sort of an in-between phenotype with both cigarette smoke and virus. And then we looked at the dividing cells. These are the cells that are really representative of those stem cells, and they reflect the same thing, is that there's more dividing cells when there's cigarette smoke alone, not with virus, and sort of an in-between phenotype with both. And so we also wanted to know if cells were dying, and this is a stain CC3, cleaved caspase 3 for dying cells. And we saw actually very little dying cells with the cigarette smoke exposure that we had given to the cells on purpose, really, because we didn't give much cigarette smoke. But we saw that in the setting of virus with cigarette smoke, there were a lot more dying cells. So clearly the injury together is causing cell death. So we were really interested in what might be going on, given that cigarette smoke together with the SARS-CoV-2 virus caused a lot more infection in our cultures. And so what we did is to do single cell RNA sequencing, where we looked at the gene expression patterns within each individual cell within the cultures. And the thing that we noticed that was most obvious to us when we first looked at this was that there was a global reduction in genes expressed in the viral groups, both with cigarette smoke or without cigarette smoke. And so that was interesting to us. And it's shown here in this heat map where you can see these paler columns, which is really the majority of the genes, because this is just a list of all of the thousands of genes that we looked at. And here we're seeing just this reduction in genes that occurred in the setting of the viral infection. But there are other groups where we saw other interesting kinds of patterns. So for example, when we looked at the genes that are upregulated in the setting of virus alone, but downregulated in the setting of cigarette smoke with virus, what we found is that there were a lot of these interferon response genes. And this is a very important innate immune response. And so that gave us a clue as to what might be going on, as to why there might be more cells that were infected in the setting of cigarette smoke with the virus. And it's known that cigarette smoking reduces the interferon response. And as I pointed out, this is a very important part of how the epithelial barrier responds to infection and how it evades infection. And so at least one of the things that's going on, we think, is that the cigarette smoke is reducing this innate immune response. It's reducing the interferon response. And that's how the virus is able to get in more easily and cause more infection. And so to look at this even more closely, what we did is to actually treat with interferon beta in our cultures. And what we found is that we could actually completely abrogate the infection. So we completely stopped the infection, even in the setting of cigarette smoke, where we saw so much infection. We completely stopped that just by adding the interferon beta 1 and really inducing a good immune response in the epithelium. And this is in comparison to remdesivir, which of course is in the clinic and being used, where we did see a good response too, but it wasn't as good a response as interferon beta. And so clearly the cigarette smoke is having this innate immune response. It's reducing the ability of the cultures to fight the infection. And so I just want to stop there and acknowledge my amazing lab members who, despite all of the restrictions that were imposed on us with the pandemic, have done an amazing job really trying to carry these studies forward. I want to acknowledge Viti and Gus for being wonderful collaborators and doing all of this work in the BSL-3 facility here at UCLA, and Catherine Plath and Justin Langeman, who did all the analysis of the single cell RNA sequencing. And I also really want to acknowledge TRDRP for their funding, because they gave us a seed grant to do this work. And without their funding, this wouldn't have been possible. So thank you so much for your attention. I'm happy.
Thank you. And next up, Dr. Glantz.
That was a great presentation. I have one question that we can talk about later, and that is, have you looked at vaping and whether it has similar effects? But I'm going to present an overview of some of the epidemiological disease evidence and on smoking and COVID, and also just briefly reinforce what you just heard. There's a pretty big literature now showing that smoking makes COVID worse, that this is a meta-analysis. Oops. This is a meta-analysis that we published about a year ago of the first 19 studies. And these are people who've already been diagnosed with COVID, looking at, you know, were they at increased risk of hospitalization or death or admission to the ICU and intubation. The endpoints varied, but overall, once you were infected with COVID, it about doubles the odds of having a more serious course. And since then, there's been a lot more papers published. We have an updated meta-analysis, peer review that has, I think, 47 papers in it. And the general conclusion holds. And also, there's an increased risk of death. There's now enough data to show that. This is from the updated meta-analysis, which is in peer review now. And we plotted the odds of disease progression against how old the patients in the studies were. And again, this is people who were already diagnosed with COVID. And we found a trend toward more serious impacts in younger patients. And in this case, younger isn't teenagers, younger is people in their 30s and 40s. But in general, they had the adverse effects of smoking were higher in younger people. Why? Well, you already heard some of the reasons in the very elegant presentation that preceded me. But smoking damages cilia. It has adverse effects in the upper airways, which affect the ability to capture and expel viruses and bacteria. The previous talk was really focusing on the upper airways. There's also lots of data that smoke depresses the innate immune function deep in your lungs and makes cells more susceptible to getting infected. It causes pulmonary inflammation, which also increases susceptibility to infection. And significantly, e-cigarettes have similar effects on the respiratory tract. And there's not a lot of data, epidemiological data, looking at the effects of e-cigarettes. But if you look at the biology, you would expect them to have similar effects to smoking. This is just a summary slide or summary picture from a really excellent review of the literature on the effects of smoking on a receptor site called ACE2. Receptors are sites on cell surfaces that you can think of as doors or windows that let things outside the cell into the cell. And the ACE2 receptor is the doorway that COVID uses to get into cells. And there's a lot of evidence that when you smoke, you upregulate the ACE2 receptors, you generate more doors that the COVID virus or the coronavirus can use to get into the cells. And interestingly, when people quit smoking, the ACE2 count goes down, and this effect is reversible. And there's a lot of epidemiological evidence to support this. And this review paper I've cited here by Smith et al., published last year, also goes through a lot of the kind of molecular and cellular evidence that you heard about previously. And, you know, an interest, this isn't in my presentation, but an interesting finding that my pulmonary colleagues here at UCSF have found, looking at both smoking and vaping, is that the levels of exposure, which don't seem to have much effect on cells under normal circumstances, have a big effect if you expose them to an influenza virus or a pneumonia virus or pneumonia. And I think that's consistent with the previous presentation, that smoking actually makes the pulmonary system more sensitive to these invasions. So what about the risk of infection? As I said, there's very broad and consistent evidence that once you're sick, COVID or smoking makes COVID worse. You would expect the risk of infection to be impacted also for the reasons that I just talked about, also for the reasons that were discussed in the previous presentation. And there is good epidemiological evidence that if you smoke, you're at increased risk of getting infected with the flu and MERS, which is another coronavirus infection. But the evidence on risk of infection is generally pretty muddy, and most of the studies have actually shown a protective effect of smoking. The argument that's been made is that the nicotine has anti-inflammatory effects that are somehow protective, but there's a ton of other, even if that's true, and it may well be true, there's a ton of other bad stuff in smoking that has adverse effects on the pulmonary system that would lead you to expect increased risk of infection. Also, smoking causes the risk factors for COVID, heart disease, lung disease, diabetes, and a long list of other problems. So there's also good reason to think there would be an indirect effect of smoking on COVID risk. But as I said, most of the studies show lower reported smoking rates among COVID patients. I think there's a couple of very serious methodological problems with these studies. One, and this is a theme of this overall conference, is the medical system generally doesn't do a very good job of assessing smoking. So there's a lot of people who are probably falling to, a lot of smokers who are falling through the cracks. And especially in the early part of the pandemic, most of the studies were very sick patients in acute distress, some of them who'd already been intubated and couldn't talk. And so it was very hard, even if you wanted to try to get a smoking history, which generally wasn't done, it might not even be possible. And there's a tremendous sampling bias in that most studies are limited to people who are tested for COVID. And because testing is still not done among everybody or a random sample of the entire population, that introduces a sampling bias in the studies. And it makes, I think, the estimates of the association unreliable. We've actually tried to estimate the magnitude of that sampling bias, but based on and which direction it goes, but we've not been able to figure out a way to do it based on the available data. There is one population sample out there, which wasn't just people who are already getting tested or already sick. And that's the study that Bonnie Halpern-Felcher's group did, which she alluded to in her presentation. And this is of youth and young adults aged 13 to 24. Was a population sample from an online survey. And they found that e-cigarette use and dual use with cigarettes was associated with substantially higher risk of having a COVID-19 positive diagnosis. Cigarettes only didn't reach statistical significance, but as you heard in the earlier presentations, among these young people, there are very few cigarette only smokers. They're using e-cigarettes or dual use with tobacco. I actually asked Bonnie to go back and calculate what the risks would be if they limited their sample just to the people who'd been tested. And these risks went way down, which is probably reflecting, what the fact that sampling bias in the other studies is a problem. So, so far, this is like the only good population sample I'm aware of. And it's a cross-sectional study, and you always have to worry about making causal statements in cross-sectional studies, but it certainly suggests a substantial risk of infection, and that's exactly what you would expect based on the biology. So the bottom line of all of this is that we need better assessment of smoking and passive smoking too, because there is some evidence that passive smoking is associated with increased risk. And we really need to be also asking about vaping and marijuana use, including vaping marijuana, because if you look at at what's in marijuana smoke, it's not that different from what's in tobacco smoke. We need, good population samples to determine the infection risk. And, one place we could have probably gotten that is these big randomized trials that have been done for the COVID vaccines, because they are population samples, but I haven't seen anyone talking about reporting smoking in those studies. But so I haven't gone looking either. There's really, very little doubt that smoking makes COVID-19 worse, and quitting smoking and vaping is an important element of COVID-19 prevention, certainly prevention of serious disease, and probably prevention of infection, although we're still waiting for good large samples to show that. So that's my presentation.
We're going to switch gears from the biological and epidemiological to now the population public health perspective. And it's my pleasure to help represent what's been really a team effort, a whole year-long effort for this California campaign to quit for COVID. And it's a great model for how research and public health partners, and many of you are on this call today, so everyone stepped up to take action and join forces, as we say, in what we all know has been an unprecedented year. I don't know how many times I've heard that. But we have some great news to share after a whole year-long effort about this, and how we've been able to support California smokers to quit during a very stressful year, and during a time when no one is going to see the doctor or wanting to go outside to go to the hospital if they can help it. So I just want to acknowledge and appreciate the many partners on this California Tobacco Control Program, led by the branch chief, April Raessler, and their media team. They really made this happen. TRDRP, especially Norval Hickman, who gave us the green light to do a lot of this. Of course, the California Smokers Helpline team, and also my California Quits team. We are a statewide project funded by the California Tobacco Control Program to help facilitate health systems change with safety net health clinics and health plans that serve the Medi-Cal population. So why Medi-Cal? It's 1 in 3 Californians and also represents 40% of California smokers. So if you all remember, it seems, I don't know, yesterday, but it's actually almost a year ago when we had the shutdown happen, right? So my first thoughts were, what's going on with people at home, you know, tobacco use. We know that there are risks for smoking or vaping. It's been described really well by Valerie Yerger earlier about, you know, there's many triggers for smoking and stress and anxiety being amongst them. And then also reasons for exposure as well. You know, it's not just whether or not you use tobacco, it's also that maybe you're living with someone who uses tobacco, or maybe you're living in multi-unit housing where someone's using tobacco. And our, you know, as we all know, our whole lives have been upended, a lot of uncertainty. You know, what it kind of changes the way we're even thinking about people's use and exposure in this past year. So, you know, one thing that I did as a doctor, I was kind of like, okay, people aren't going to be coming to see people like me now. We really need to turn to public health and try to figure out how to message and support people at this moment. And so the California Tobacco Control Program has a public-facing website called tobaccofreeca.com. And April Raisler and her team were super supportive in saying, look, you know, we can put up a message, you know, trying to explain to people why now it's a great reason to quit for COVID. And, you know, we were seeing that message already on social media. So we worked together on just putting a little doctor message with 3 reasons to quit now. As you've already heard, we already knew smoking doubles your risk of developing respiratory infections. That's from the Surgeon General's report. And that ranges from anything from colds to tuberculosis. And then we were seeing growing evidence, like from Stan's study, that smoking doubles your risk of getting sicker from COVID-19. So, you know, getting intubated or risk of dying. And then, of course, we had just come off the EVALI epidemic, where we saw that vaping was also associated with with new diseases for lung injury. And so as the evidence about vaping's lung harms grows, you know, we had just finished coming off this EVALI epidemic, so to speak. I mean, it's still continuing. There are still cases. The CDC just stopped tracking. But, you know, definitely a lot of reasons to protect your lung health as soon as you can. And then, as I mentioned, you know, there's also this concern, you know, the majority of the population are non-smokers, and we're all, you know, at home now, especially if you think about vulnerable populations and in public housing, you know, what's going on with people stressed and maybe smoking inside. So this was something they put up also, where I said, hey, can we just even have something like this for, you know, approaching a stressed out neighbor who's smoking. And if you want to go to this, I still like this idea of saying, hi neighbor, you know, please take care of yourself. And, you know, I'm here to help you however I can. And some of you who work on tobacco control might be amused by this, but you know, one of their strategies is just to insert smoking amongst many other things. And this is one way to at least approach wellness. The California Smokers Helpline did an amazing job posting up a COVID webpage when we started talking all together about how can we all work together to help support smokers. So if you go to nobutts.org/COVID, you can see what their team put up in terms of saying, look, this is a time to take care of your health, and how do you cope with stress and nicotine withdrawal, which if you've ever seen someone in nicotine withdrawal, you know, it's really bad in the first few days. You know, you get highly agitated or irritable even, which, you know, maybe folks don't realize what's going on and how to cope with that. And then we approached TRDRP also and said, what can we do with our research grant? Because people weren't coming through the clinics as to LADHS, which was our primary project. And Norval Hickman, our program officer, was so supportive and said, you know, and agreed that we could help turn some of our research money around to offer free nicotine patches to all Californians. You know, previously the Helpline and I had worked on the Medi-Cal Incentives to Quit Smoking project, which offered statewide free nicotine patches. They have a first 5 special offer, but this is for all Californians. Our California Quits team with Rebecca on our team worked with some of the priority population projects funded by the state tobacco program to try to tailor some materials to some of the groups that we know already have higher tobacco use rates. So if we still have this available, and I will give you a simpler URL about how to download some of these materials from our website. And this is a limited time offer still, it's still going on. And you know, every few months, every quarter, we're reassessing about should we be continuing this because it is a big lift and a lot of funds, but we think that it's well worth the effort. So stay tuned. And in summer 2020, you know, it took a few months, but the California Tobacco Control Program's media subcontractor started synthesizing this info and putting it together as a Quit for COVID ad campaign. And these are examples of some of the digital ads that came out. And the first one's on the left, you can see that's packed with a lot of info, and then some of the ones on the right, which I've been seeing more now, a little bit more simpler message, just reminding people, you know, you got to protect your lungs with COVID. And, you know, it was great that we got all that stuff started off because it took a few months also for approvals for the California Tobacco Control Program to put up this great website under the Department of Public Health. And so you can also go to this website and look for more information to link to. And I believe it's the Bonnie and the Stanford Toolkit team that also put this great 1-minute video demonstrating why it's so important to protect your lungs with COVID-19 right now. And then in fall of 2020, I surveyed a bunch of our California Tobacco Control Program partners. You know, the program had already said, hey, there's all these resources, promote the helpline, promote Quit for COVID. So we did a survey of respondents, and we had quite a number of local lead agencies, which are typically county public health departments, and also funded projects. And amongst all these respondents, it reflects the majority of California. Thank you to everyone who helped answer this survey during the time of COVID, when a lot of people are also being pulled for other responsibilities. And it was just amazing to see that the majority of them had reported promotional activities, whether communicating this information about quitting for COVID, or posting information about tobacco treatment with the helpline, doing in-person education, doing additional media on top of what the state program was doing in terms of media. So really at the local level, at the county level, you know, tailoring it for populations. Very grateful for everyone who's contributed to this effort. And the folks who didn't do promotional activities, you know, they were just getting funded typically. You know, the state tobacco program funded a lot of new projects that were just getting up and going during the COVID pandemic. So this is something very exciting to share also. This is the state tobacco program has been doing quarterly mailings to all Medi-Cal households. 7 million Medi-Cal households. They've been doing it for several years now after we've completed the Medi-Cal Incentives to Quit Smoking project. And so again, this is something great where Norval supported us, where we said, hey, could we see what kind of messages might be effective out there? And let's also put a message comparing free patches versus free quit service. And then also other flyers saying, you know, quit for COVID versus stop smoking. So this is called a 2x2 study, but basically it's 4 flyers. They're in English and Spanish, double-sided. And it's this was sent out to throughout October to December of 2020. And it's just sharing some prelim results. It's been an amazing caller response, and we're even seeing a bigger caller response with the free patches. So taking that prelim result, knowing that the free patches message was so powerful, we are now doing another round of this Medi-Cal mailing, January to March, where we are saying stop smoking and also free patches. And so this is expected to go through April 2021, but like I said, we're kind of assessing to see how much longer should we be continuing this for all Californians, not just Medi-Cal, but all Californians. And we're able to track the response because there's a promo code that the helpline asks people for when they call. And I'll mention this coffin ad in a minute too, again, because it apparently is a very powerful ad that the state tobacco media program has noted from before. So great news, we've had over 11,000 callers from the helpline receive these TRDRP nicotine patches, and it's just a 2-week starter kit, but it's mailed to people's homes directly within a couple days after talking to the helpline. And so this is just a bar chart showing you how many people have been helped over, over 10 months. And the helpline does have more nicotine patches from other offers that were provided first. So this is just showing you the picture from TRDRP funded patches. And this is the great news slide where, we just managed to take a step back from all this and say, well, what's going on with people quitting and with the helpline doing all this work? And so the blue line shows all the calls from 2020, over 20,000 people who called the helpline and, and did their intake. So not just people who called and said, oh, actually, I don't want to, partake in services. These are the folks who, made it through and asked, and we have information about. And then this is a 20% increase from 2019, which had over 16,000 callers. And that was, you might wonder, what is that spike in the orange? And that is the coffin effect, I think, from that JBR mailing that quarter to the 7 million Medi-Cal households. Again, we'll see what happens in 2021 when we're sending out that image, and people may be taking note of it. By the way, those mailings, it comes with a whole bunch of other stuff. It's a mandatory mailing from Medi-Cal for all benefit information. So not really an exciting kind of mailing, but maybe that image is actually taking people's notice and people are paying attention to it. So in summary, our California Quit for COVID campaign, I think it's been an amazing partnership all around, especially with the different media channels we've had to use. And this is supporting all of us clinicians as we're trying to have people not get sick and not come to the hospital, to try to quit at home. And we've had some great success so far, showing an increase in the helpline calls. And again, many thanks to all of you for helping to promote this, and please continue to promote this. You can get the flyers at our caquits.com website, and we're hoping to reassess, see if we can continue promoting maybe in the next quarter past April. We'll see how much funds we have. So I will pause there, and maybe if we have any time for discussion. Thank you. So just from skimming some of the questions, I may just clarify. I believe that's for 18 and up. It's only for cigarette smokers. Right now, there's not a nicotine dosing protocol for vaping at the helpline, although it's much needed. And as I mentioned to folks before, though, teens can go to their provider and get plan covered medications. It's not a contraindication. It's just that when we're talking about the randomized trials, we don't have a lot of scientific evidence to say this is the formal recommendation of treatment, but certainly people going into who are using nicotine products and are highly addicted, they can go to their provider and get products as well. And the TRDRP funded nicotine patches, if you just call the helpline will offer it to all callers. They will ask about any potential contraindications. For example, if you had a recent heart attack, then they will say they will need to talk to the doctor before distributing nicotine patches, or if you're pregnant, for example, as well. So there is a protocol that they follow, and happy to answer more questions.
Okay, there's a couple here. One for Dr. Gompert. Do you think you will find similar findings in lungs in non-smokers exposed to secondhand smoke in COVID?
Yeah, we So we don't know for sure, because obviously, though we're exposing directly to cigarette smoke, we can't really quantify how much it is. But because we gave such a low dose of cigarette smoke, just 3 minutes a day for 4 days, we think that it could potentially apply to people who are having secondhand smoking issues. So I think it could translate. And then to answer the vaping question, that is something that we're very interested in. Like I said, we can expose our cultures directly to vaping, to the aerosols from vaping, and we have been doing this. We have some interesting findings that are taking, I think, longer to figure out, because the trouble with the vaping is that there is so many thousands of different brands and so many thousands of chemical combinations. And so what we're finding is that the basic PGVG nicotine base causes some toxicity to the airway epithelium, but it's really the flavors and probably the heating of the chemicals in the flavors that are causing the majority of the toxicity. And so to study each of those in combination with the virus, that's a lot of work, because we're also only able to vape one flavor at a time. So one of the things we're trying to do in the lab, actually with TRDRP support, is to try and be able to get to a more high throughput system where we can test maybe 20 or 50 different vapes at a time, and then compare them across the board in something of like a toxicity screen. So that's the difficulty right now.
Okay, great. And another question for you, do your interferon findings have clinical application in treatment of COVID-19 infection?
Yes, and I just in the chat privately, thanks to Bruce, who told me about some nasal interferon studies in basically in SARS-CoV, sort of more like common cold type viruses, where there have been found to be some positive results. I will say that giving, like as a clinician, I know that giving like IV interferon, it's very toxic. It gives you really bad flu-like symptoms because you really are driving that innate immune response, and that the IV studies that I'm aware of have actually not shown to be a benefit. So maybe not. I think it's still too soon to tell, but I think maybe there'll be other therapies that will improve the innate immune response that could be helpful. I actually have a question, not being an epidemiologist or sort of being more involved in the basic science level, but I was sort of struck by looking like at the WHO website at how the reports have been coming through about maybe cigarette smoke is protective against COVID-19, which is really going against what we've seen, and how much misinformation is out there. And I wonder what you feel about in terms of strategies about how to counteract this misinformation. I mean, obviously, other than doing like really great prospective studies, but what could we do right now?
Well, this is Stan. I mean, I've actually worked with the WHO on that stuff, and it's a huge problem because the tobacco interests have a huge PR machine. And for example, as I said, there is some reason to think that nicotine by itself might be protective by downregulating inflammatory responses. And a person in France, who incidentally has a history of working with the tobacco industry, announced he was going to do a study of that. I mean, they were very careful in the announcement to say we're just going to study it, and they were looking at nicotine patches, not inhaled nicotine. But that got covered all over the world as if they had found that smoking was protective. And as I said, there's a huge sampling bias in basically all of the studies out there, because they're just looking at people who are tested and not looking at a population sample, except for that one study of Bonnie's. But it's the old problem of, a lie can get around the world 5 times before the truth gets its shoes on. I mean, the WHO and others are trying to counter that, but it's a problem. I mean, we really, need good data, and we need, people to be asked, about their smoking and vaping, including marijuana, and we need to get it at a population level. But again, because COVID testing has been so limited, just because of, throughput, it's been, very hard to get asymptomatic people in any kind of random way to get tested. So it's a big problem. But everything in the biology points to whole smoke being a problem and increasing infection risk. I mean, I haven't seen a single study of whole smoke which shows a protective effect. And the nicotine studies are iffy. But for example, work done by our pulmonary group here at UCSF, looking at cigarette smoke and e-cigarettes and susceptibility of the lungs to infection. This is looking at the alveolar level, both in whole animals and using preparations similar to the one you used, except for alveolar cells. Have found that that the nicotine does blunt the adverse effects of the smoke and e-cigarette aerosol, but it doesn't make it go away. But especially when you get into these things where there are multiple competing factors, explaining that to the public, especially when you've got an industry out there promoting their point of view, is a real problem. In fact, this morning, there was a report in Bloomberg News that Philip Morris has put a formal request into FDA to start advertising that there's nothing wrong with nicotine.
Oh,
So, but again, I mean, it's just hard to believe. And the one little thing we did, as I mentioned, is we asked Bonnie in her study to go back and recompute the risks if they limited, rather than looking at their entire sample, was to just limit it to people who got tested. And the results did move, in the opposite direction. So I think there's, we need good population level data. We just don't have it. For infection, once you get infected, there's a large literature showing adverse effects of smoking.
And maybe the problem is that that message hasn't necessarily gotten out to the public. I feel like it's been hidden probably by the tobacco companies, saying that smoking may be protected, but they don't say anything about if you're a smoker and you get infected, about how much more likely you'd have to severe disease. That people don't seem to understand this.
Well, no, I agree with you, and I think part of this is another area where California has really been, taking a tremendous lead by running the campaign that Eliza talked about. I mean, you're just not seeing anything like that coming out of the FDA or the CDC or any of the national health groups. And I think that the people in California and the California Department of Public Health have done a really good job of threading the needle on this and making scientifically accurate statements that people can understand, and sort of sidestepping some of these, complexities.
Do you think that we should be prioritizing vaccinations for smokers? I think some of the states have been doing that, but I haven't heard California talking about that.
Well, some places are prioritizing smokers and also people with obesity, because obesity is pretty clearly a risk factor. I think, I mean, I'm not an expert on COVID vaccination strategies, but the fact that, I mean, it's not irrational to prioritize smokers. I mean, there's certainly people that shows that the policymakers think smoking is a risk factor. I think fortunately the Biden administration has done a pretty good job of ramping up the availability of vaccines, and they're becoming widely enough available that problem will become moot in terms of vaccinations.
okay, we're getting close to time. One last question from Dr. McDonald. Are there any plans to look at quitting success in the helpline callers from this campaign? I guess that's probably for Dr. Tong.
Oh, yes, the helpline does look at a small sample of their smokers annually and call them back to see if they have quit. Just the magnitude of this project, we're not going to be able to sample everybody, but we will have some results. And the California Tobacco Control Program, I learned from April anyway, that they're going to be sharing some information on population-based tobacco use rates, and it's been looking very good for California. So again, thank you to everybody for helping to help all Californians in this time.
Thank you again, panel, for such great information that you've shared with us.